So we're going to finish up the lecture on the stomach. Where we're at is we are talking about the innervation and circulation in regards to the stomach. So if you guys recall from what we learned in the circulatory system, there off of the descending aorta, there is the celiac trunk. And so then that celiac trunk here is going to branch off, and there's going to be the hepatic branch, and one of those branches is going to be the gastric artery, and that's where the blood supply is going to come from. Blood will be drained from the stomach and the intestines as well, and that will drain through the hepatic portal. All blood from the digestive system is going to be filtered through the liver before it can enter into the venous system and go into the heart. We're also going to have some nervous stimulation. The stomach will receive parasympathetic nerve fibers from the vagus nerve, and it will have sympathetic fibers from the celiac ganglia. So if we dive way back to 201, we learned just off of the vertebrae there was some sympathetic celiac ganglia fibers. That's what's going to provide sympathetic innervation to the stomach. The movement of the stomach or gastric motility is going to actually begin with swallowing. So when a bolus is swallowed, that swallowing center is in the medulla oblongata. And that is going to send a signal to your stomach to start to relax. So that way, when you swallow that bolus of food, it will travel down the esophagus and it will arrive in the lower esophagus. And that lower esophageal sphincter will open because the stomach has now relaxed. The vagus nerve is what's going to relay the message from the medulla oblongata, and it will activate a receptive relaxation in the stomach. So there will be this brief stretching, but it will end up relaxing so that way the stomach can hold more food. Soon after the stomach has received the food, it's going to start to contract those muscles. And it's going to start these waves of peristaltic contractions. And those waves of contractions will be controlled by pacemaker cells that are within the longitudinal muscle layer within that muscularis externa. So remember, that's the most superficial layer of muscles. And we'll have a ring of constriction that occurs about every 20 seconds. And those contractions become stronger and stronger as they approach the pyloric region of the stomach. After about 30-ish minutes of these contractions, they get pretty strong. And that contraction that's happening in all three of these muscle layers is going to churn the food, and that churning is going to physically break it down, and it's also going to increase the surface area, mixing that food with the gastric juices, and that's going to promote the chemical digestion along with that physical breakup of the food. Once that chyme is churned, it's going to start to get released into the duodenum or the duodenum. I say duodenum, that's how it was taught to me. Tomato, tomato. But when we're talking about how much, it's not all released at once. There is about 3 mils of chyme that's released at a time. And just to put that in perspective, 5 mils is a teaspoon. So less than a teaspoon of chyme is going to go into the duodenum at a time. And this small amount is beneficial because it's going to allow the stomach acid to be neutralized. Sodium bicarbonate is going to get released from the pancreas, and that's going to be able to neutralize that stomach acid, which would otherwise burn through the intestinal walls. And then it will also help us digest, or help the duodenum digest nutrients little by little so we can get the most out of that food substance. Also, if the duodenum does start to get overfilled, it will begin to inhibit gastric motility. So that way the food will stay in the stomach. And a typical meal will empty from the stomach in about 4 hours. If the meal is mostly liquid, like a soup, it'll be a little bit of less time. But if it's a really high fatty meal, it could be around 6 hours. Sometimes when we eat, the food doesn't stay down, and we undergo this very unpleasant experience of vomiting. And vomiting is a forceful ejection of the stomach contents. And if there's a very forceful ejection, it can even include some of that intestinal content as well. And that ejection is going to come from the mouth. So, in order to induce vomiting, there's actually a lot of coordination that has to go into this by the medulla oblongata, and it's going to integrate a lot of different muscle actions. Some of the things that can induce or cause vomiting would be overstretching of the stomach or the duodenum. There could also be some chemical irritants like alcohol or toxin. There could be visceral trauma or even intense or physiological sensory stimuli could induce vomiting. Before vomiting occurs, there's usually some nausea involved and retching. Retching is that I'm not feeling so good action that happens before the vomiting. So during retching, there is actually an expansion in the thoracic cavity and the abdomen contracts, and that puts a pressure difference that will start to dilate the esophagus. And then this dilation is going to cause the lower esophageal sphincter to relax, while the stomach and the duodenum are going to start being kind of spastic with their contractions. And chyme will leave the stomach and go into the esophagus, but it doesn't leave. It gets blocked by the upper esophageal sphincter, and then it goes back down. So it's not full vomiting yet because it goes back down, but it's definitely that feeling like, oh my gosh, I'm gonna puke. And it's usually accompanied with tachycardia, some salivation that's going to prepare and lubricate the tract for that vomit, and sweating. Vomiting will occur when the abdominal contractions get enough to overcome that thoracic pressure, and it forces the chyme through the esophagus and past the upper esophageal sphincter. Then the esophagus and the body of the stomach will relax, and the chyme will be driven out of the stomach in the mouth by these strong abdominal contractions. And that is also going to be combined with some reverse peristalsis of the gastric atrium and the duodenum. Chronic vomiting can cause some problems. For example, there can be very dangerous fluid levels and electrolyte and acid-base imbalances that develop. Bulimia is an eating disorder, and the hydrochloric acid in the vomit will cause the enamel in the back of the teeth to start to erode. There is also a danger of aspiration. That is when some of that acid gets inhaled and gets into the respiratory tract. That can be very damaging to those bronchioles and to the delicate epithelial tissue in the alveolus. And sometimes surgical anesthesia can induce this feeling of nausea. So that's why a lot of times they have you fast to make sure that the stomach is empty and that the small intestines are empty. So that way it kind of lessens that feeling for vomiting and not retching. The stomach is going to play more of a primary role in the digestion. Most absorption of nutrients is going to happen in the small intestines. And that will occur after the chyme has passed into those small intestines. It does not itself, the stomach, absorb really any significant amount of nutrients, but it can absorb aspirin as well as some other lipid soluble drugs. The hydrochloric acid is going to activate a lot of those salivary enzymes like lingual lipase, and then there's some gastric enzymes like the pepsinogen being converted to pepsin, and the lipase that's secreted in the stomach. Those enzymes will start to get activated and partially digest some of those proteins and lipids. And a lot of people might say, well, okay, alcohol is absorbed in the stomach because why is it that when people eat, they tend to not get as drunk or they don't get as drunk as fast? Well, alcohol is absorbed by the small intestines, but the intoxicating effects will be dependent on how empty the stomach is. Because if the stomach is empty, then more of the alcohol will get put into the small intestines to be absorbed versus a full meal where we're going to have just a little bit of the chyme released at a time, which would have the alcohol in it. The stomach is going to have some protection against that hydrochloric acid. There are 3 ways that it's protected from the acidic and enzymatic environment that it creates. The first one that most students are pretty familiar with is that mucous membrane. It is very thick and it's actually alkaline, so it can resist the action of the acid and a lot of those enzymes. And then those cells, the epithelial cells, the surface mucus cells, they are going to be really tightly packed together, and they're actually going to have these structures called tight junctions, which connect the cells together, which will prevent gastric juices from seeping in between them and getting into those deeper tissues. And then these cells have a very high mitotic rate. That means that they're going to undergo mitosis and they're going to replace themselves very quickly. For example, your skin cells replace themselves every 28 days, your liver cells a year. These cells are going to be about every 3 days. They're going to get sloshed off with the chyme and they're going to get digested with the food, and they will be replaced by cells that are doing mitosis within the gastric pits. And if these defensive measures don't stay intact, we can have ulcers form, and it results in inflammation, and they can be pretty painful. So if we look at this picture here, we can see this ulcer start to form, and it's showing you where the acid from the stomach is now starting to eat away at that stomach lining. It's digging into the mucosa layer and the submucosa, and then it can even get into that muscle layer. So gastritis, which is inflammation of the tissue, can lead to an ulcer. But most ulcers are caused by a bacteria that's resistant to the acidic environment known as Helicobacter pylori. And you can actually see it here. This is that bacteria. And usually it resides in the pyloric region of the stomach. And it is treated with antibiotics and Pepto-Bismol. When it comes to regulation of the stomach, we will have the nervous system and the endocrine system work together. If we have an increase in gastric secretion and motility, that's going to happen when food is eaten. And when the stomach empties, that's going to be detected by these systems, and we will start to see a decrease in that gastric secretion and motility. Gastric activity can be divided into three phases. There's the cephalic, the gastric, the intestinal phase, and these phases we'll talk in the next couple of slides in more detail, but they overlap and they can occur simultaneously. So the first phase is the gastric phase, and this is where the vagus nerve is going to stimulate gastric secretions to begin. So this can happen before the food is even swallowed. We are smelling it, we're seeing it, maybe we're chewing it so we can feel and taste it. In this period, the food is also then swallowed, and semi-digested proteins will also start to activate gastric activity in the stomach. About 2/3 of the gastric secretions is going to occur during this phase, and about half of the acid secretion in the stomach is going to happen at this phase. When food is ingested, that is going to also stretch the stomach, and it's going to kind of flatten out those rugae. So that will stimulate the gastric activity, and it will activate what's called a short reflex through the myenteric plexus. And then it will activate a long reflex mediated through the vagus nerve and the brainstem. And so that long reflex is going to be what tells your brain, hey, I've had enough to eat. But it takes about 20 minutes for that reflex to get to the brain. And then we can also increase this gastric activity by increasing the pH of the contents. If we have a high pH in the stomach, that tells the stomach, hey, I've ingested some food, I'm not so acidic now, so I've got to kick up some gastric activity. I've got something in my stomach. Gastric secretion is going to be also stimulated by three different chemicals. We have acetylcholine that can be secreted by our nerve fibers that are in both the reflexes, the short and the long reflex. And then histamine will have a paracrine effect on those cells when they are secreted from enteroendocrine cells in the gastric glands. That will stimulate gastric secretion. And then also a hormone called gastrin. Within the pyloric section of the stomach, those enteroendocrine G cells will release gastrin that can stimulate gastric secretion. So in the second phase is called the gastric phase. This is when food stretches the stomach, and it's going to activate that myenteric and vagovagal reflex. And these reflexes will stimulate gastric secretions. And then histamine and gastrin are also going to stimulate acid and enzyme secretions. The third part of the phases is the intestinal phase. And so this is when the chyme is going to arrive in the duodenum. The duodenum will respond to that chyme by moderating gastric activity by using hormones and nervous reflexes. So the duodenum will initially enhance the gastric secretions. Briefly, it's going to stimulate the stomach. The stretching of the duodenum will accentuate the vagovagal reflex that stimulates the stomach. The peptides and amino acids in the chyme will stimulate some G cells that are in the duodenum to secrete more gastrin that's going to further stimulate the stomach. However, shortly after, acids and fats will trigger the enterogastric reflex. The duodenum will send inhibitory signals to the stomach through the enteric nervous system. So secretins, CCK, and the enterogastric reflex will inhibit these gastric secretions and the gastric motility while the duodenum is processing the chyme that it already has in it. The sympathetic nerve fibers will suppress the gastric activity while the vagal or the parasympathetic stimulation of the stomach is now inhibited. And the duodenum will also send signals to specifically in the medulla to inhibit vagal nuclei. So that reduces the vagal stimulation of the stomach, and it will stimulate the sympathetic neurons that send inhibitory signals to the stomach. Chyme present in the duodenum also has an effect on the enteroendocrine cells that are present in the duodenum. And it causes these cells to release secretin and cholecytokinin. Both of those will stimulate the pancreas and the gallbladder to start releasing substances, and it will suppress or inhibit the gastric secretions. So we're going to have a decline or a decrease in how much gastrin is secreted, and the pyloric sphincter will start to contract very tightly, and that will limit the chyme entering into the duodenum. So that way the duodenum can have some time to work on the chyme that's already in it. Those enteroendocrine cells will also secrete glucose-dependent insulin-tropic peptide, or GIP. That was originally called gastrin inhibiting peptide. That's where GIP comes from, but we still use GIP as the abbreviation. Well, GIP will stimulate insulin secretion and start to prepare for the processing of these nutrients, and specifically glucose, that's about to enter the bloodstream from the small intestines. And so here, this picture is showing that there is actually two different stimuli for these G cells to secrete gastrin. It will be secreted both in the presence of the protein and in the presence of food. You can see the neck mucus cells. You can see these Pac-Man looking cells are the parietal cells. And then we have the chief cells here. And then these kind of triangle ones are those enteroendocrine cells. And so we're going to pause it here, and we'll talk about the liver, the gallbladder, and pancreas next.